Hello, everyone, and welcome to the GC for All webinar. It happens every third Thursday of the month. This webinar is hosted by me, John Lewick, at the University of Rochester, and Rick Cooley at the GCE for All Center at Oregon State University. The webinar features exciting research being done in the GCE space and also those that are GCE adjacent. We've had talks that focus on incorporation of novel, non-canonical amino acids to their novel uses in cell biology and all the way to protein translation termination processes. And so it's kind of a wide net that we cast here. While we have had prominent and established investigators in the field present their research, we've also want to provide an opportunity for early career scientists to share their work. For example, just senior graduate students that are about to defend their thesis and are looking for postdocs, and postdocs who are about to go on the job market and are looking for either academic or industry jobs. So please send us suggestions of future topics that you'd like to hear about, and also speakers that you think would like to be highlighted in this forum. So today we have two great speakers. We have Satpal Virdi from University of Dundee, UK, and Toshi Teramoto at the National Institute at the University of Tsukuba, Japan. First up, we have Toshi Teramoto. Toshi got his bachelor's and master's of science in material science from Osaka City University. He then went on to Tokyo University of Agriculture and Technology to get his PhD in engineering. There, he worked with Tetsuya Asakura on the engineering of functionalized silk for both industrial and biomedical applications. He's currently at the National Agriculture and Food Research Organization in Japan, where his research focuses on using genetic code expansion to install amino acids that allow for click chemistry and molecular level functionalization of silk actually in the silkworm. He's received several awards along the way, including two Young Scientist Awards in 2011 and in 2015 from the Japanese Society of Sericultural Science, a Best New Frontier Award in 2016, and most recently in 2025, a Japan Silk Society Award for his work in this space. And he's been a real leader in developing technologies to work in the silkworm, which I think is a pretty unique space to be in. So with that, Toshi, take it away.
Yeah, thank you. Thank you very much for kind introduction. And as I share the slide, you should, maybe you can see the slide.
Looks
And
perfect.
okay,
Yep.
but it's okay. So thank you. Thank you very much for kind introduction. And my name is Toshi Teramoto from Nara, Japan. And today I would like to talk about GCE as a platform for producing functional silk fiber materials. I think the fibrin is not familiar with most of you. And so in this case, fibrin is the major protein component of silk fiber produced by silkworms. So the reason why we are focusing on the sorry, uh, sorry. Okay, sorry. The reason why we are focusing on silk protein is that it is renewable and naturally derived protein material with outstanding mechanical strength and excellent biocompatibility. So that is why silk fibrin has been widely studied for biomaterials development. So next, let me start with some background on sericulture in Japan. So I think sericulture is not familiar with most of you, and sericulture is a kind of agriculture involving the rearing of silkworms like this to produce silk fibers. And historically, Japan was a major silk producing country in the early 20th century, but the industry has rapidly declined in this almost 100 years. And today, only around 100 farms remain in Japan. So this situation clearly represents a crisis for the industry, right? But at the same time, it creates an opportunity to redefine sericulture itself, not as a traditional industry, but as a new type of protein manufacturing system. So traditionally, silkworms were reared in open environments in farmers. So in contrast, the silkworms are rare. Usually, recently, some factory-based sericulture has emerged in Japan over the past decade. In this system, silkworms are reared in a closed, highly controlled environment. And this enables stable, reproducible production and effectively transforms cell culture into a manufacturing platform for high-value protein materials, which is fully aligned with sustainability goals. So then, why is this level of control so important? And actually, factory-based sericulture provides a highly controlled environment, which is better for the reliable production of medical-grade silk materials, which, for example, for use for the some regenerative medicine or something. And at the same time, closed environment is compatible with genetically modified silkworms, which must be under strict human control, right? So to fully exploit these advantages, control must extend from the production system down to the molecular level design. So conventionally, silk protein design has relied on the genetic engineering at the DNA level. For example, researchers have generated fluorescent silk or hybrid silks with spider like this, or bagworm silk sequences. But even then, we are still limited to the 20 canonical amino acids, So, but in contrast, GCE works complementarily at the translational level, which allows us to introduce amino acid analogs and greatly expand the molecular level design space. So here I show some history of my research. Our group has developed GCE systems specifically for the domesticated silkworm, Bombyx mori, which has been used for the sericulture for thousands of years. The system was established step by step, starting from the engineering of aminoacyl tRNA synthetases, or ARS, then moving to the cultured cells, and finally to direct incorporation into silk fibers produced at the individual level. So this achievement was one of the few reports of GCE conducted with multicellular animals, and even now the examples of multicellular animals is very few, as you know. And after this initial success, we achieved a substantial improvement in the incorporation efficiency through collaboration with Dr. Sakamoto at Riken, and leading to our key publication in 2018. And here I show our method. Our GCE system employs a residue-specific method, not the site-specific method, because the residue-specific method is relatively simple and just requiring engineered ARS mutants. And also it allows the incorporation of multiple amino acid analogs, which is particularly suitable for material applications. So today, I will introduce 3 repositories of silkworm GC systems. And here the key idea is to select target amino acid based on their abundance in silk fibrin and the intended functions. So for example, the low abundance of phenylalanine is ideal for chemical functionalization without affecting the physical properties. On the other hand, tyrosine is much more abundant and is suitable for tuning physical properties. And the third pyrolysin system has been studied to enable further expansion of the amino acid repertoire. So first, I introduce silkworm GCE targeting phenylalanine. Here I show the system for incorporating phenylalanine analogs like this. And we generated mutants of phenylalanine tRNA synthetase, or PRS, in collaboration with the Riken group. And very importantly, we expressed the PRS disappearance mutant only in the silk producing gland. So this red part inside the silkworm larva, and by using the established transgenic techniques. So this organ-specific expression avoids adverse effects on the whole body of silkworm, such as growth inhibition. So actually, we tested several phenylalanine analogs, and among these analogs, we mainly focusing on the azide-bearing analogs of shown here, and the name is azide phenylalanine, which can be used for the functionalization by click chemistry. So when the transgenic larvae were fed this azide phenylalanine mixed into the diet, we were able to harvest cocoons composed of silk fibers incorporating azide phenylalanine. So the expression of different VRS mutants shown here resulted in different replacement ratios in silk fibrin from phenylalanine to azide phenylalanine, and also the different production of silk fibrin. And based on these results, we selected the first one, this mutant, as the best among those tested. And this structure model shows the position of phenylalanine in silk fibrin indicated by red stars. And the phenylalanine is very rare in silk fibrin, only 35 residues out of about 5,500 amino acids. It's very, very large protein, you know. And even with roughly 7% replacement, this corresponds to only 2 to 3 azide phenylalanine residues per silk fibrin molecule, which means less than 0.1%. So the incorporation is very, very low. And importantly, this very low incorporation does not significantly influence the physical properties of silk. So just make changes on its functions, you know. And so these azide groups enable click chemistry, as you know, and specifically azide alkaline cycle addition, and to make a stable triazole bond. So because this reaction is bioorthogonal and highly selective, we can introduce a wide range of functional molecules, such as proteins, pigments, drugs, and polymers, and anything else without damaging the silk backbone. So this concept forms the basis of what we call clickable silk, which is the name of this special silk containing azide groups. And using this clickable silk platform, we demonstrated some bioorthogonal modification of silk materials. And so selective click reactions allow functionalization exclusively at the azide groups, even in the different material formats, such as fiber, film, and sponge-like materials. So you can see clear fluorescence only from clickable silk, the green and red ones in the fiber, film, and sponge, and not from the control normal silk. So it means these results show the stability of azide groups during processing. Sometimes the fiber is once dissolved in a solvent, but the azide is still stable and can be reacted by click chemistry. On the other hand, these azides decompose upon UV radiation. So by using this feature, we can create precise patterns of functional molecules on silk materials, even at the micrometer scale. So here I show one application of clickable silk to control cell adhesion on silk surfaces. Basically, cells attach very well onto the silk surface. However, by connecting the PEG chain, polyethylene glycol chain, by click chemistry, silk surfaces became cell repellent, as shown in this figure. And by combining with UV radiation technique, we can spatially control cell adhesion on the silk surface. So look at this picture. The left part of this picture was UV irradiated, where you can see good cell adhesion. In contrast, the right part was not UV radiated, where PEG chains were attached by click chemistry. So you can see that no cells in this right part because the surface became cell repellent. As a result, you can see the very clear boundary of cells, even in the same silk substrate. And you can see like specific patterns made of cells. So this is a very interesting application of silk, clickable silk, And next application is the use as a drug carrier material by mobilizing fluorescent molecules as drug models through a linker molecule having a photocleavable structure. So we demonstrated their stimuli responsive release upon light irradiation. So you can see clear decreases of fluorescence from only from the areas of light irradiation. In the case of film and textile, you can see the almost no fluorescence on the irradiated areas. So this concept of stimuli responsiveness can be probably extended to different stimuli, not only by light or, for example, by heating or by some magnet stimuli or something, by designing appropriate linker structures here. So next we move on to the GCE targeting tyrosine residues. Tyrosine is much more abundant than phenylalanine, occupying around 5% in silk fibrin. And tyrosine is found in the highly repetitive crystalline regions of silk fibrin, which is responsible for the physical properties of silk fibers. So by incorporating tyrosine analogs like azide tyrosine or some halogenated tyrosine, we tried to directly influence the intermolecular interactions, such as cross-linking or molecular packing. To achieve the incorporation of tyrosine analogs, we engineered the tyrosyl tRNA synthetase, also in collaboration with the Riken group. And we used the structural information from the human counterpart to guide the mutation designs. And this selection system has led to the selection of 4 mutants of TRS. And we have generated transgenic silkworms expressing each mutant. And after feeding experiments, we found that the first one, these mutants, was the best among these 4 mutants. So using this mutant, we generated silk incorporated with azide tyrosine, and we found that the mechanical strength was improved. And even though the replacement ratio was very low in this case. And actually, mechanical testing showed a slight increase in the stress and the toughness of silk fibers. And we suppose that this improvement is attributed to interchain cross-linking at the azide groups. On the other hand, the incorporation of halogenated tyrosines improved the thermal stability. The replacement ratio was larger than that of azide tyrosine for both halogenated tyrosines. And this DSC data showed an increase in the decomposition temperatures of about 5 degrees centigrade by the incorporation of this amino acid. And so these examples of tyrosine analog incorporation, and these changes probably comes from the molecular packing by the introducing halogen atoms. It is a larger atom, which makes some packing effect among interchain, among the silk protein chains. So these two examples of tyrosine analog incorporation demonstrate the molecular level modifications can translate into the changes in macroscopic properties of silk fibers. So I think it's a very interesting applications of GCE to make some hyper functional protein materials. And finally, I introduced the pyrolysis system. So the pyrolysis system allows access to additional amino acids bearing large side chains like these. And in addition, the target amino acid in silk fibrin can be flexibly changed by altering the anticodon of the pyrolysyl tRNA. So in this case, the anticodon was set to glycine because it is most abundant amino acid in silk fibrin. And by incorporating this TCO lysine, we can successfully modify the silk fibrin using Diels-Alder reaction. So this system significantly broadens the design space for silk materials. And these are the amino acid repertoire that have ever been incorporated into silk fibrin. So we are now trying to further expand the repertoire. And in the last part, I briefly want to introduce our efforts toward the industrial translation of clickable silk, maybe for 5 minutes or so. And we first improved the silk production by conventional breeding techniques because the transgenic silkworm is very small, so it should be improved. And after improvement, we use this new line for test production of clickable silk in a closed facility owned by a private company with up to 10,000 worms. So clickable silk was successfully produced and processed into textile products like these. And of course, the reactivity of azide still remained. And one application we are now pursuing is antibacterial fabrics in which antibacterial agents stably immobilize via click chemistry. So as we move toward commercialization, a major obstacle lies in the high cost of azide phenylalanine, which is very expensive, And so we are now addressing this by increasing incorporation rate and also exploring intracellular biosynthesis from cheaper precursors like this. So in this first challenge, we anchored the TRS mutants to the ER membrane inside the silk gland cells, which results in preferential incorporation of amino acid analogs into secreted proteins such as silk fibrin. And in this second challenge, we are now trying to elucidate and engineer amino acid metabolic pathways. So these cost reduction efforts remain a key challenge for us. So we are now seeking collaborators for practical applications, and for that purpose, we exhibited our test products like this at the exhibition held in Tokyo last year. And so our current target products include silk-based antibacterial, healthcare, and medical fabrics, especially for sensitive skin. So this is a summary slide, and GC provides a powerful platform for designing silk materials at the molecular level beyond conventional genetic engineering. So we do believe that this approach represents a new paradigm for sustainable protein-based materials manufacturing. And finally, I would like to acknowledge my collaborators and partners and funding agencies who made this work possible. And thank you very much for your kind attention. Thank you.
Thank you, Toshi. That was wonderful. If there are any questions, you can ask them on the chat and we will try to get to those, or raise your hand. You can ask them in person. I'll start off with the first question. So you kind of alluded to this, that your incorporation efficiency is very low, and you're doing a couple tricks to improve that. But it also seems that even with low incorporation, you have significant change in the physical entity that is the cell. So what is your ultimate goal? Like, what do you think you can achieve if through modeling or through any sort of kind of just thinking about it, I guess, improvement in tensile strength? Do you think you can improve it tenfold, or is this like, what do you think the ceiling is?
Yeah, tenfold would be wonderful, but probably is very difficult. And actually, we are now trying to maybe follow the properties of synthetic polymer fibers, like polyester or nylon, which has a little bit higher tensile strength than silk. And also, the such synthetic polymer has a lot higher temperature. I mean, uh, such polymer can be used in higher temperature than silk fiber. So if silk can be more tougher and more thermally stable, and silk can be used as a replacement of such synthetic fibers, for example, as filler of some plastics or something
Yeah,
to make the materials stiffer. So that is the our goal to make silk fiber compatible with such synthetic polymers.
great. Is that Paul? You have a question?
Yeah, that was really interesting.
Thank you
I was
very
just
much.
wondering if you mentioned that you can chemically functionalize the clickable silk,
Mhm.
and that involves sort of dissolving the protein in a solvent system. Is that what you mentioned? And I
Uh,
was wondering, yeah, how do you do that? What is the solvent system, and what are the implications
yeah, yeah,
in
yeah.
terms of cost? Because you've got to, extract the silk back again, and
Uh-huh.
does that affect the,
Mm-hmm.
integrity, the structure of the silk once it's been through you know,
Mm-hmm.
the
Maybe
dissolution?
you asking about these processes?
Yeah,
Oh,
you mentioned that. Yeah, so you
yeah,
mentioned
yeah,
that
yeah.
you
I,
can
I,
click
I
on
said,
like pigments and things like that.
mm-hmm, mm-hmm. Yeah, silk can be dissolved in some solvents, and so this is a really established technique to make different materials. So even much more types of silk materials can be fabricated from the such solvent system. And so this is also an already established technique. So I said that even through such conventional fabrication techniques, the azide still remain its reactivity. So that is very interesting for us to make these any types of silk materials can be used for further functionalization by click chemistry. So that's a very interesting point for us.
Okay. And just one more question, if I may. So
Hmm.
you
Absolutely.
mentioned that the azidophenylalanine results in cross-linking,
Hmm.
and that's UV-induced photo cross-linking, right?
In the case of phenylalanine,
Yeah. And,
yes,
and is that happening in vivo or during the biosynthesis, and do you irradiate the
I
worms
see. I see. I
to
see.
that, or how is that controlled? And
I'm I'm not sure about the difference of phenylalanine, like the case of phenylalanine and tyrosine, even though they both have the same functional groups, azide groups. But in the case of phenylalanine, we didn't see the any cross-linking, and but in the case of tyrosine, azide tyrosine, we
yeah,
saw the cross-linking during the processing, when the cocoons are processed into fiber, we need some processing to make the other proteins should be removed, and some heating must be necessary for the processing. And in that process, the azyl tyrosine uh, probably decomposes and makes some cross-linking in the closed other chains, silk chains. But in the case of phenylalanine, the similar thing doesn't happen. So probably it is due to the number of amino acids, or maybe in the position. The phenylalanine only exists in the linker region, which is not in the repetitive region. So, and on the other hand, tyrosine residue present in this crystalline region. So maybe the secondary structure and tertiary structure of proteins is different. So probably that makes such difference, I think.
okay, thanks.
Thank
Andy,
you
do you
very
have a
much.
question?
Yeah, and there's one in the chat. Do you want to ask that
Yep.
first?
Yeah, I'll ask the one in the chat first. So, from Praveen Singh, does AZF modification and clickable silk provide hydrophobic or hydrophilic properties for drug administration.
Probably, I think it's possible. And yeah, actually, I show the case of PEG modification, which makes the silk surface much hydrophilic, and that is why the cell cannot attach onto the surface. Probably, if we use some different molecules, such as long alkene chains or something, we can see some hydrophobic surface. I think we can create probably, but I have never tried that.
Got it. Andy?
Just quick. You said with 35 fees and about 7% incorporation, you get 1 to 2 modified phenylalanines maybe per silk chain. I'm wondering, do you do any post-analysis mass spec degradations to figure out if maybe there are what the ratio of different phenylalanines get incorporated? So maybe there's 2 or 3 that are preferentially incorporated and the other ones not at all. Or do you have any sense of the mixture of molecules you have?
Actually, we always analyze the mixture of molecules, so we don't have any method to analyze only single
Perfect. You
molecule.
haven't broken it down and figured out which phenylalanines are modified.
Yeah.
Okay. It's a big molecule.
And so actually we treat fibers or some materials, which of course it contains a much amount of molecules. So probably the number of azide groups will be probably averaged in the materials.
And I have a follow-up question for that, actually. Do you think that the system is self-selecting? So do you think you'd have higher incorporation, but you just won't get silk production out of the silk gland? And so do you think in some instances you've changed the physical properties of the silk to the point where the worm can't make it? And then only when it leaves the gland can you make the modifications?
Yeah, very good question. Yeah, I also have the same question. And sometimes, yeah, in the case of genetic modification, so if we modify the silk gene drastically, sometimes silkworm cannot spin the fiber or even cannot make the proteins. So in my case, in the GCE, the replacement ratio is still low, so that such thing doesn't happen. But maybe if the incorporation rate becomes higher, maybe such thing will happen. I'm not sure, but
yeah, the gland will give you the can't do this sign. Yeah, that's really interesting. We should move on to the next speaker. Thank you so much, Toshi, for a great talk.
Thank you very much.